Welcome to the fourth talk from the ISMNIC talk series, In Love with Addiction Neuroscience. I am Parnian, one of the members of the executive team of ISM Neuroscience Interest Group. Before starting the talk officially, I just want to mention some housekeeping notes that you should bear in mind while being in this meeting. So the first thing is about the video recording of this session. The meeting will be entirely video recorded and it will be shared publicly on the ISMNIC YouTube channel. So if for any reason you wish not to be in the recorded video, you may want to keep your webcam off during the webinar. And yep, that's it. I hope you enjoy the whole talk and everything.
Good. And for just the first kind of 2 minutes of talk, if everybody who is online right now, great. Rita, the floor is yours.
Okay. So good morning, everybody. Well, at least in the East Coast in New York, it's still a morning. It's my great pleasure to be here today and to introduce Jean-Lud Cadet, a senior investigator with tenure and the chief of the Molecular and Neuropsychiatric Research Branch and Section at NIDA, Intramural Research Program. Jean-Lud Cadet, MD, came to the US in 1970. He attended Brooklyn College and then Columbia University College of Physicians and Surgeons, where he obtained his MD in 1979. He did his residency in the Department of Psychiatry at Columbia University in New York, and then in the neurology department at Mount Sinai Medical Center, where I work, actually. So that's our shared history there and presence. And then he went to NIDA IRP in the Intramural Research Program in 1992, when he's now is the chief of this branch and section. Dr. Cadet has co-authored numerous papers, abstracts, book chapters in the hundreds, and his impact in the field is very well felt. Any measure you look at is extremely high. And he's been studying the mechanisms of neurobiological mechanisms of addiction with a focus on cognitive dysfunction, observing cocaine and marijuana abusers, and actually the work that influenced me a lot, he did also with Karen Bola, and that's some of the work that when I just started studying addiction, it was extremely influential. The purpose of his research is to identify cellular and molecular mechanisms that underpin stimulant and other addiction, including epigenetic and transcriptional changes in the brain. Presently, his lab is studying how stimulants and oxycodone can cause changes in the expression of genes and certain proteins in the brain. And he's also investigating the epigenetic basis of meth and oxycodone addiction by using animal models of drug self-administration. So here the focus is on behavior. His lab has shown that unexpected food shocks can separate rats who are already taking a lot of methamphetamine into addicted and non-addicted rats, with differences attributed to expression of ion potassium channels in the brain of those animals. And this has been shown to be similar in his lab also to oxycodone-taking animals, and with similarities to the human literature. The overarching goal of Dr. Cadet's work is to try to develop better treatment for humans who abuse psychostimulants and or opiate drugs. He received numerous awards. I'm going to just mention a couple of the recent ones: a NIDA Diversity Mentor Award and an NIH Director Award, the Harvey J. Bullock Award. And that's it as an introduction, and I'm very much expecting towards your talk. So thank you very much, everybody, for attending, and I'm sure we'll have a treat.
Thank you very much. I want to thank the organizers for inviting me to participate in this series of talk. The speakers are who's who in addiction research. So this morning, what I'm going to be trying to do is give you a historical perspective of what I've done in addiction research, starting with my time. Okay, so this is what my talk is going to be like. So I'm going to talk to you about when I grew up in Haiti, talk a little bit about when I first came to the United States, and then medical school, and then most of the focus would be on the number 4, which is myself at NIDA in Baltimore. So a lot of people don't know anything about Haiti. And as you can see here, Haiti is very close to the United States. It's in the Caribbean islands. It's close to Cuba and Puerto Rico. And Christopher Columbus supposedly discovered Haiti in 1492. And then after Columbus got there, many of the native indigenous people in Haiti basically got killed. And then the Spanish brought slaves, and then the Haitian fought a war of independence against French. And then this is the Haitian flag. Sometimes the top of the flag is black, sometimes it's blue, depending on who's running the country. So it's really an interesting story about the Haitian flag. So this is me as a baby and my family. This is really a bad photograph from many years ago. And this is my grandfather. So I can tell you that my grandfather was much more famous than I ever will be. He was a Haitian painter. And then this is one of his paintings that I have in my house. This is actually my last class in Haiti. I went to a Catholic school, and all of these guys, it was a very selective high school. Most of these guys are now in United States or Canada. So, and several of them are also physicians. This is my brothers and my father, and I'm sure you can guess which one I am. So this is me. And so now I came to the United States in October 70. This is me in Brooklyn. And I should tell you about this. I wrote my first poem after arriving in the United States because it got really cold that first winter. And as you know, Haiti is pretty warm. I've never seen snow, never been that cold before. I thought I was going to die because it was so cold. So after that, I went to medical school. I moved to Manhattan. This is Central Park. I'm sure some of you have seen it. And I went to medical school at Columbia, like you were just told, and did residency. And then I moved to NIDA in 92. When I moved to NIDA in 92, I just came from Columbia as an assistant professor. And having lived in New York for a long time and also going to college there, during my college years, I met a number of young people who were using a lot of marijuana. And then, so they were freshmen together with me, but over a 2-year period, I noticed that some of them actually, the heavier users, were no longer functioning well in school. So, and then in medical school again, Columbia is very close to Harlem in New York. Doing my training as a medical student, my training as a resident, I also saw many people who were abusing drugs. At the time, heroin was a very prevalent drug of abuse around Harlem. So I encountered a lot of people who suffered from substance use disorders. So when I came to NIDA, I was very curious to find out what happens in terms of exposure to drug. So the first thing I wanted to do was look at the neuropsychiatric, behavioral, neurological aspect of substance use. In this presentation, I'm focusing on marijuana use disorder first. So when I wanted to do that, I'm not a neuropsychologist. I was able to set up a collaboration with Karen Bola, who was at the time an assistant professor of neurology at Johns Hopkins Bayview. Together, we started this protocol at the intramural program of the NIH NIDA. And so the reason why we started with marijuana was the most commonly used drug, as you all know, in the world. So I wanted to know, and I had to say that I have many friends in college who were using marijuana. And in the United States, there are 2 to 3 million new users per year. And the majority of users are when they start, between age 12 and 17. Obviously, in college, most of those guys were above 18, and 1 of 12 new users become addicted to drug. Actually, this is a point I'm going to come back to in terms of the basic science that we're doing now. Not everybody who uses a drug becomes addicted to the drug. This is very important to keep in mind. Okay, so the first paper we were doing was to look at cognitive function in marijuana users. So the specific aim of our study was to determine if there were existence of a cognitive dysfunction and if those dysfunction would persist after we admitted the participant for 28 days on a research unit. And then we wanted to determine if this deficit were dose-related, and then we used joints per week as the dose. So that's the marijuana group. The participant had to claim marijuana as their drug of choice. They had to use marijuana for at least 2 years, smoke marijuana at least 3 times a week. That criteria I opted for because some of the people I knew in college were basically using marijuana almost every day, and others were using it maybe not ever less than 3 days. So I thought that would give me an indication of who it is who might develop cognitive dysfunction. And the rest, they had to be HIV negative and spoke English. So we admitted the participant onto our research unit, and they had access to no drug whatsoever because it was a closed unit. And then we tested them after they were on the unit for 3 days, and then we tested it again at the end of their stay on the research unit. And this is the battery of neuropsychological tests that we use. And so this is the breakdown of the groups of marijuana users. And then we were able to separate the marijuana users into light, middle, and heavy users. And as you can see on the figure here, depending on, we had a specific cutoff in terms of how much marijuana they were using. The one thing that was surprising to us was in the heavy group, the amount of marijuana some young people would actually use. So even with my experience in college, I didn't expect that there would be so many, that there would be individuals who would use so much marijuana. And then, so this is the Wisconsin cat sort. And as you can see here, the more marijuana somebody uses, the more cognitively impaired on the Wisconsin cat sort they are. And then in terms of motor function, finger tapping, again, the heavy group was the group that was the dysfunctional group. As you can see, the light and medium group, they actually tested similar to control group. So in summary, then what we found that when we compared the 3-day testing to the 28-day testing, we found that The participant had problems with verbal memory, executive functioning, visual perceptive function, psychomotor speed, and manual dexterity. So after a 1 month of abstinence, this dysfunction still lasted. So that paper, we published that paper, and right now that paper has been quoted more than 500 times. So we're very happy about having done this. So at the time that we're collecting data on marijuana users, the protocol was open to people who were using cocaine. In Baltimore, I should tell you, there are a lot of crack users and heroin abuse in Baltimore. So we're able to enter a number of patients who meet criteria for cocaine use disorders. We basically did the same thing that we did with marijuana. We brought the participant in to the ward. We tested them at 3 and 28 days, and then see if they had any deficit. Now, the inclusion criteria for this study were that they needed to have used cocaine by any route, minimum use of 1 year, and they had to have used cocaine 4 times per month, and they also had to have a urine toxicology test positive for cocaine metabolites. And we excluded other folks who were using, say, heroin and some other drugs. Again, this is the group, and then, so as this is the demographic of the cocaine users, and you can see that the patients were using a lot of cocaine. Again, when we look at all of the neuropsychological tests, we saw significant differences between the cocaine users and the control group from Baltimore. So the one important thing that I think people who are interested in neuropsychology of substance use, you have to use a control group from the same environment so that the exposure to other stress and other issues like that can be matched. So most of the subjects in this study were African American young men, and then we were able to recruit young African men from Baltimore so that they could be matched on several demographic criteria. And again, as with the marijuana group, heavy users of cocaine had more trouble. If you look at TRELS-B, look at CALT, speed of information processing, and some other tests, the heavier group showed the most cognitive impairment. So in summary, then, with marijuana, chronic heavy cocaine abusers had persistent neurocognitive deficits. They had deficits at 3 days after admission and deficits at 1 month after admission. So being cleaned of drugs didn't really help much. The other thing that we found was that the higher amount of drugs that these users were abusing, the more cognitively impaired they were. So we concluded then that the cognitive deficits that we're seeing might have been due to direct effects of chronic cocaine use because of this dose-dependent effect that we were seeing. So we, a few years ago, Ahmed and I, we wrote a nice review of some of the neurotoxicological, neuropsychological deficits reported in people who abuse drugs. So my perspective is that all treatment programs should take these neurocognitive deficits into consideration when planning long-term care of patients who suffer from marijuana use or cocaine use disorders. And that's even truer for methamphetamine use disorders, because if you look at the literature, several investigators groups have published on the cognitive deficits seen in some methamphetamine patients who suffer from methamphetamine use disorder. Now, in the last part of my talk, I'm going to spend a significant amount of time discussing what we're doing now. So you can see the progression. As when I first came to NIDA, I spent a lot of time doing clinical and basic science, but in the last few years, I've focused mostly on the basic science aspect of my research. So as you know, methamphetamine use disorder is very common. When I've gone to places like Korea or Hong Kong or Japan, mostly what investigators in those places talk about is meth abuse. There's very little cocaine, and obviously marijuana is everywhere, but there's very little cocaine in some of these places, but meth use is very, very prevalent. And so in the DSM, substance use disorders, it is described as a neuropsychiatric disorder that has associated with it loss of control of drug use, craving of the drug, continued use despite adverse consequences, and compulsion to use drugs. In the case of methamphetamine use, and this is true of other drugs, there's initial exposure to the drug. The patients increase their meth dosage over time as they become tolerant, and then they go through this meth binge where they spend all of their money taking methamphetamine around the weekends and stop taking the drug until they run out of money, or whoever they're using together with doesn't have any more methamphetamine for them to share. And then at that time, there's forced abstinence and withdrawal, and even when they're in treatment, they go through this multiple relapses. So it's very important for us to come up with pharmacological and psychological treatment that would keep people in therapy and prevent relapses. So this is the way methamphetamine works. Methamphetamine gets into the monoaminergic terminals, and then it gets into vesicle and displaces dopamine that gets released in the synaptic cleft. Dopamine release in the synaptic cleft then binds to dopamine receptors that are located postsynaptic. So in our studies, we've looked at both the pre-terminal effects of methamphetamine and the postsynaptic effects of methamphetamine, and we've tried to decide which drugs are antagonists of dopamine receptors that can block the effects of methamphetamine. So this slide shows what happens when you give an animal methamphetamine. Depending on the dose, there's a huge increase in the level of dopamine in the synaptic cleft. obviously, that's one of the reasons why people like to use drugs like methamphetamine, because they get a high from using this amount of a drug. So, as I mentioned before, we're going to come back to the slides. As you can see here, a lot of people try to smoke, people try heroin, cocaine, alcohol, and marijuana. But when you look at the percentage of people who develop addiction itself, is a low percentage. For tobacco, it's actually higher than heroin, higher than cocaine, and then alcohol and marijuana. I don't have methamphetamine on this slide because I couldn't find in the literature, but from anecdotal reports, 50% of people who use methamphetamine tend to develop methamphetamine use disorders. So it was very important for us to develop models not only of drugs exposure, but of continued use of drug in the presence of adverse consequences. So we wanted to know whether it would be possible to develop rodent model of substance use disorder using food shocks at adverse consequences to produce pseudo-voluntary abstinence or continuous compulsive drug taking. Some other investigators have done this. They've done it with alcohol and they've done it with cocaine. And the amount of shock used by these investigators vary for alcohol and also for cocaine. There's several, but nobody had done it for methamphetamine. So in my lab at NIDA, we decided to try the same experiment. And in the slides that follow, I'm going to show you that the use of food shock can actually lead to segregation of methamphetamine animals that had already escalated their use of methamphetamine into shock resistant. Those are the rats that are taking the drug compulsively, they're addiction prone, or into shock sensitive rats. Those are non-compulsive, and in the case of human, with people who expose themselves to drug but are resilient and never develop substance use disorder. So we've published a number of papers now. So what I'm going to do is show you one of our better papers looking at the epigenetic and transcriptional consequences of doing this type of experiment on the nucleus accumbens. So this is what the model looks like. So the training phase, in the training phase of the experiment, the rats are put in a box where they can press a lever to get methamphetamine. And rats actually like methamphetamine. Within a few days, they learn which lever would give them methamphetamine. And after they do this for about 20 days, we put them through a shock phase where when 50% of the time, when they press the lever to get methamphetamine, they also get a food shock. Administration of the food shock lead to animals that are shock resistant and shock sensitive animals. This is the behavior experiment. As you can see here, all of the rats will escalate their use of methamphetamine. Okay, so rats really like methamphetamine. But after we apply food shock, we start with 0.18 and then progressively increase the amount, the intensity of food shock. We can separate the rats into shock-sensitive animals, animals that basically stop pressing the lever to get methamphetamine, and shock-resistant rats, animals that continue to press the lever to get more methamphetamine. So the red circles are basically humans who, even if you put them in prison, if you take away everything, all of their possession, no matter what, they will continue to abuse methamphetamine. And the one thing that we were amazed, we've done this several times, so 43% of the rats are shock resistant. So they become compulsive methamphetamine takers, while obviously 57% of the rats become methamphetamine, are shock sensitive, and they stop taking methamphetamine. So this is what happens at the end of the experiment. So this represents the last day of drug self-administration when the rats had escalated their use. So this is for the shock-sensitive rat. And then this is what happened after they are exposed to the contingent shock. So there's a significant decrease in amount of methamphetamine that the shock-sensitive rats take. But in the case of the shock-resistant animals, the compulsive animals, this is what happens last day. So they're taking a lot of methamphetamine. There's no significant difference between the groups. But here, as you can see here, there's very little decrease in the amount of methamphetamine that the shock-resistant, the compulsive rat, continue to take. And there are significant difference between the amount of methamphetamine that the compulsive rats take in comparison to the non-compulsive rats. So in our lab, what we think is going on is that open exposure to drugs, and this include cocaine, marijuana, heroin, oxycodone. We're studying oxycodone in my lab. So exposure to drugs, there are individual factors, and there are also environmental factors that influence whether somebody become a drug abuser or meet the criteria for substance use disorder or not. So these exposures we think are associated with differential changes in gene expression. And we also think that there are epigenetic alterations that are dependent on these individual factors, and these epigenetic alteration can influence changes in gene expression, and then that lead to differential changes in protein expression that drive what happens behaviorally. In order to test those ideas, we've used multiple approaches. We've used quantitative PCR, microarray analysis, and now everybody's using RNA sequencing. And then, but for the epigenetic investigation, we're using global DNA methylation, DNA hydroxymethylation, followed by PCR to see what epigenetic changes might occur in the brain of animals exposed to these drugs. So in the experiment that I'm showing you, we did that. So the control group and the methamphetamine, whether they were compulsive or resilient animals, we extract DNA, and then we do this hydroxymethylation DNA immunoprecipitation, and then we did genome-wide sequencing of the DNA, and then we end up with results where we found hydroxy DNA methylated peaks, and then we can compare all three groups based on those hydroxymethylation peaks. So those are the results of the experiment. They're showing you the result of the gene sequencing, and we can compare the compulsive rats to the control rats, and what we see is that a large number of genes have hydroxymethylated peaks when we're comparing the compulsive rat to the control. When we compare the compulsive rat to the non-compulsive rat, we see even more genes that have hydroxymethylated peaks. And then the resilient rats, they don't behave like control. As you can see here, there are also many genes that show changes in hydroxymethylation. So exposure to methamphetamine, even if it doesn't lead to compulsive drug use, you can see there are a lot of epigenetic changes that can occur in the brain of somebody who exposed themselves to methamphetamine. One of the things that we found when we were doing this study is that many of the genes that showed differential methylation between the shock-sensitive rats and the shock-resistant rat belong to families of potassium channels. So in this slide, what we did was use ingenuity pathway analysis to see how these genes were connected to other genes. And as you can see here, those genes are involved in transport of molecules, transport of potassiums, and importantly to us, they were also involved in cognition processes and long-term synaptic long-term depression, and also in Krebs signaling in neurons. So the cognition part was very important, as I mentioned before, and in the slide I showed you before for marijuana and cocaine, several groups of investigators have shown that subgroups of patients who abuse these drugs can show cognitive dysfunction. So we're very excited about this because potassium channels, as you know, they're found on axon terminals, they're found on cell bodies, and they participate in regulating electrophysiological events that occur in neurons. So we then decided that we should pursue this further. We wanted to know if changes in DNA hydroxymethylation would be reflected in changes in messenger RNA in the methamphetamine group. And as you can see here, when we measured mRNA with PCR, what we found was that there were significant increases in the expression of potassium channel in this shock-sensitive rat. So the KCNA1, A2, B, several of this potassium channel that showed differential hydroxymethylation also showed increases in the level of expression. We've measured protein expression in some of these potassium channel, and the protein expression is also affected, indicating that indeed that potassium channel might play a significant role in mediating suppression of methamphetamine self-administration. So in summary, for the study, what we've concluded is that when we use quantitative analysis of DNA hydroxymethylation in meth-addicted and unaddicted rat, we find quite a number of genes with differentially hydroxymethylated peaks in the nucleus accumbens. Prominent among those are members of potassium channel, and the expression at both the mRNA and protein level are increased in the nucleus accumbens of the non-addicted rats. This experiment identified for the first time a role for different classes of potassium channel in promoting abstinence for drug-taking behavior in an animal model of meth addiction. I should tell you that after we did the study, we searched the literature. It turns out that other group of investigators had suggested that potassium channels are involved in mediating alcohol-taking behavior. And if you use agonists at the site, you can suppress alcohol-taking behavior. We have not done this experiment with methamphetamine yet, and that's some of our plans to test whether activating potassium channel can suppress methamphetamine taking behavior. I went through this with you. What I hope you were thinking about is the fact that I came from Haiti and moved to Brooklyn, and then I ended up at NIDA. So on the basis of that, so you can ask the question, what are the odds, okay, that I, as a Haitian immigrant, moving into a Caribbean hotspot in Brooklyn, would end up at Columbia University and then at NIDA and being able to publish all of these papers. So what I want you to take from my talk is that scientific research is for everyone. As a young man growing up in Haiti, I didn't even know that you could do this kind of stuff. I didn't know that physicians did research. I thought physicians just took care of patients. So this didn't dawn on me until I was a third-year medical student at Columbia, when the professors who were running the wards where I admitted patients were talking about their own individual research. So I said, wow, physicians can actually do that. So in the fourth year of my medical school, I went to the NIMH, and I've met a number of physicians at the NIMH who were doing research. So that was what sold it for me, and I became a researcher. I should also tell you that you need to find mentors who will support whatever it is that you want to pursue. When I first came to the intramural program at NIDA, I did a presentation to all of the section and branch chiefs, and several of them were very critical of my plan to study the behavioral neurology or neuropsychiatry of substance abuse. I remember one of them saying, this doesn't happen. These drugs are not associated with any cognitive impairment. But luckily at the time, the scientific director, when I talked to him afterward, he said, this is actually your budget. You can do anything you want to do with your budget. So it doesn't really matter what the other section of branch chiefs say. It's your budget. So I was able, thanks to the scientific director at the time, pursue my research interests, and thanks to Karen Bola also who was willing to participate with me in this venture, we ended up publishing quite a few papers together. And so finally, I say, don't surrender. Never surrender when you're pursuing your dream because things take time. Thank you very much.
So thank you very much for a wonderful talk. I have so many questions, and thank you for all the recommendations for everybody who wants to do research. I cannot agree more with all of these recommendations. Let me start with the last question I had, and then I'll open it to everybody. But it was so interesting, and it reminded me the work that really had so much influence on me because there was nothing else in the literature, except for your work with Karen in that early days of thinking about cognitive function in addiction, which was entirely new thinking. So you definitely were one of the people who saw it and the importance of it to study addiction in humans and then animal models. So I was thinking, when you were presenting those animals that were resistant versus susceptible to shock, the methamphetamine exposed animals, did you have the chance to actually measure their cognitive function? So any perseverance type of behaviors in those who were resistant to shock, like reversal learning type of paradigm, something animal equivalent to the Wisconsin?
So that's a very, very important question, Rita. So we've discussed this on several occasions in the lab because of all of the people in the lab actually are aware of our obvious clinical studies. So that's part of our plan. Modern studies of the brain of these animals, but to actually study them cognitively. So we intend to do that. You may be aware of the fact that we published a paper recently in psychopharmacology in collaboration with some other people at NIDA and National Institute of Aging, where we've published some experiment in methamphetamine animals. But those animals were not split into shock sensitive and shock resistant. So in that paper, there were cognitive deficits in the animals that were exposed to methamphetamine. So we intend to do the same type of study with this new paradigm. So this paradigm is novel, so we haven't had time to do all of the studies that are needed so that we can better compare those animals to human use disorder patients.
Yeah, when you were saying those are compulsive animals, I wanted to see data showing that. That would be, it would advance the field and connect both very disconnected fields in my mind. Another question I had was, in your earlier human studies, did you have a direct comparison between the cocaine and marijuana users? Did you ever do that? Remind me if I forget.
So what we didn't do that. We still have the data. We could probably go back and do that. That's a very important point. What we did was look within groups, right, as to whether people who use less or more drugs showed more deficits. We did one study where we compared cocaine patients who didn't use alcohol to cocaine users who also use a lot of alcohol. So when we did that comparison, the patients who use cocaine plus alcohol showed more deficits than the patients who use only alcohol, only cocaine. So it would be interesting to compare cocaine to marijuana. But, it's really interesting point where we never thought of doing that. I don't know why we didn't, but we had the data. That's a good point. But we still have the data.
And we're thinking now about comparing, is it an addiction general type of symptoms, or is it something that's unique to every drug? It's still interesting nowadays. We still don't know enough about that.
No, we have the data. That's a very good point. So I'll have to get our biostatisticians in a building to look at those data. That's a really important point. Thank you.
No, thank you. So let me open the floor now. I see a question, but maybe people can, either I can read it or people can just tell it themselves and ask the question. So there is a question from Milad Ravanbankash. I'm sorry. I'm not sure how to spell it out, how to pronounce it. Oh, here, Milad, tell us how to pronounce your last name, and then you can ask the question.
I'm Milad Ravanbakhsh. It's hard to say. I agree. Hello, everyone. Thank you, Dr. Cadet, for your inspiring and profound presentation. I really enjoyed it. I have a question concerning parallel concepts which may present these adverse consequences you mentioned in psychological or cognitive terms. Do you think self-criticism or self-conscious negative emotions like shame and guilt can be taken as a self-punishment strategy similar to that put shocks for rats? And if so, then could it be that sensitive people or people with insecure attachment would be more likely to develop addictions following this kind of inner punishments and criticism?
Yeah, that's a very important issue in terms of what does psychological stress do. We haven't done any study related to this, but Bruce McEwen, who died recently, his group, they've actually looked at the impact of this kind of stresses, emotional stresses, psychological stresses, and looked at epigenetic changes that happen in the brain of animals. I mean, they've done it in animals, obviously not in human yet, and they've shown that there are significant changes in epigenetic markers related to mitochondrial function in the hippocampus of animals that were exposed to various psychological stress. We've actually, because of those experiments, we are actually looking at what the effect of the adverse consequences in the absence of drugs. Okay, what are the impact of just stress on the brain? I mean, this is a very, very important issue actually, because you know that people who are exposed to various stress in the environment, whether personal or sociological, have more of a tendency to develop substance use disorders than those who are not exposed to this kind of stresses. So I think it's a very important question that you asked, but we haven't done those studies yet, but other investigators. I mean, it's a good, really good literature to look into, because those papers were published in some really important journals like Molecular Psychiatry, Proceeding of the National Academy of Sciences, the papers from McEwen's group. So that's a very important question.
Those others who are online, it would be nice if you turn on your videos, seeing faces from people around the world who are active in the talk right now. May I make a question?
Yes, please.
Jean-Lud, I was just kind of looking inside the really exciting presentation that you have made in terms of the difference between the shock-sensitive and shock-resistant rats. I was wondering, do we have any sort of predicting algorithm to be able to predict which rat is going to be shock-sensitive or shock-resistant based on their kind of their information before getting into the paradigm, and how much the algorithm could be successful in doing the prediction?
So one of the things that we did in the other study was to do a cognitive function test and some other behavioral tests before we exposed the rats to methamphetamine. I mean, we could do something similar here where we put the rats through a number of tests and then see how they behave. But I can tell you, these rats, they all came from the same vendor. They grew up together. They had nothing really. When we order the rats, they usually come, basically, they're siblings. So we think there are genetic differences that you may not be able to detect behaviorally. So we, even though we're thinking that might be an important thing to do, what we want to do is after we accumulate enough animals is to do sequencing of the genome of those rats to see how different they are. So in the present study, we show potassium channels are important. So what we want to know is that when we do the genetic sequencing, whether there are differences in potassium channels that make the shock sensitive animal more likely to suppress their methamphetamine intake. But if you look at the slide, basically the animal escalate their use. It's escalation, and then if you went to college in the United States, there are a lot of young men and women who escalate their use of marijuana. But as you saw on that slide, only a small percentage of those folks develop marijuana use disorders or cocaine use disorders. So genetically, there might be differences, but behaviorally, we may not be able to document what those differences are using available tests. I don't know what Rita thinks about that. Do you think there are psychological tests that you could use to predict?
Well, yeah, I'm thinking about the work of the group from Cambridge that also has this resistance to shock in addition to two other criteria for addiction. And those animals that show the addiction type of behaviors, including resistance to shock, are impulsivity. So impulsivity predicts who becomes addicted. In their work, I don't remember if it was with methamphetamine or cocaine. The rats that were more impulsive, and they tested them on behavioral paradigms, they had higher chance of becoming addicted and also relapsing after abstinence. So that's exactly the type of work I was thinking that really resonates so well with your work.
So in terms of impulsivity, so we think that's a thing because many investigators have shown that. So what I was thinking, the reason why we didn't go after that, it was like it's really basically while we replicating what many people in Europe, in the United States have already shown, that impulsivity is very, very important. So we didn't really want to go after that. And then we were more interested in, okay, the exposure happens, all the animals escalate. So what happens epigenetically or transcriptionally that make this animal different? And then what is the genetic basis of this? So the reason why it's important to know what the consequences are is that you may develop a treatment where you can impact the changes in hydroxylation or the changes in gene expression. So that's where our focus have been. I don't know too many good manipulation that people have used to influence impulsivity in humans.
Jean-Lud,
Let me read some more. Maybe we can go back to that discussion, but there are a few more questions that I thought I'll read out, or if Ken Winters, if you want to read or to say your question. If not, I'll then the table of views. I'm sorry?
I'd like to see the faces like Ahmed say while they ask their questions.
No, it was just, I was just going to, I have a kind of something like a final concluding question. I will ask question after other students or others who would just ask their question,
So I'll just read it verbatim. The table of use and risk for addiction is interesting. Any thoughts on the risk for co-addiction for the most common drugs of abuse? And any evidence that epigenetic changes in the rats are transfected to future generations? So that's a question from Ken Winters. Two questions from Ken Winters.
Okay. So we haven't done the studies with the pups from pregnant mothers, but there are groups in Boston and elsewhere using cocaine and opioid, and they've demonstrated that the behaviors of then adolescent animal are impacted if they've been exposed to drugs in utero. I think that's definitely an issue because, as you can see here, even though we're showing epigenetic changes and gene expression changes in the brain, methamphetamine goes a lot of places. Okay, so side effects, adverse consequences for methamphetamine use include cardiac problem, lung problem, kidney problems. So it's definitely getting into sperms and other places in the body. So those could impact the lives of future generation. Then about the comorbidity issue, so we're doing the same type of experiment with oxycodone. So what we want to do in the future is see if animals that are resistant to shock or sensitive to shock, after we expose them to either oxycodone or methamphetamine, if they develop the same type of patterns for the other drug, right? So we'll go from methamphetamine to oxycodone and from oxycodone to methamphetamine, go through the same paradigm. That way we'll know if the same genetic or transcriptional or epigenetic changes are responsible for comorbidity. Right now, you should know that there are a lot of people who are using heroin are also using methamphetamine and vice versa. The comorbidity of opioids and methamphetamine is at an all-time high. So that's not really very good. And we need to do something about this.
And I have another question from Alireza Valian. Alireza, would you like to ask it or you want me to read it? Or?
Thank you for the great talk. Hi, everyone. You were just answering the question that Dr. Kerry has asked. And I think I found it you telling that among the young men and women in the United States that are using marijuana, I mean, not all of them are addicted to marijuana. And you said that they are genetically different but behavioral the same or something like that. I didn't get it right.
So the last part of the question?
Did you say that the difference between the people who became addicted and do not became addicted, or the difference at the genetic level or at the behavioral level or something like that?
Oh, okay. So that's a very good point. So I think that if you look at the rats that we're studying, and if you also look at the numbers in humans, I think the differences are going to be genetic and environmental. So in one of the slides that I showed, I point that exposure to drug impact the individual, but exposure to drug also impact the interaction of that environment, of the individual with that environment. And the environment can influence gene expression, okay? So that's one of the reasons why we're doing studies where we're just manipulating the environment, say with food shock, to see what are the impact of the environment on the brain in terms of epigenetic, gene expression, and protein expression. And as I mentioned a few minutes ago, Bruce McCune's group has shown the impact of psychological manipulation in rats, hippocampus. So I think the environment is very important, but I think the environment is working on a genetic predisposition. so that if there were not the environmental influences on an individual, it's possible that that individual would not have developed substance use disorders. And if that individual was not exposed to drug, that obviously that individual would never develop substance use disorders. They might develop some other psychiatric issues, okay? Because I think all of these psychiatric disorders are intertwined. So, but in terms of substance use, I think both the environment and the individuals are very important.
So, Jean-Lud, the follow-up question to this is potency. In human studies, it's really hard to control for potency. And you did it so nicely, dividing the marijuana users to heavy, medium, and light, and the cocaine users also based on days and amounts of use. But with marijuana in the 70s, let's say 80s, and now, there are such huge differences, right, in their actual potency. And in animal studies, if you add that component to all the mix you just mentioned about the interaction between the stress and the genetic endowment and the drug itself, did you think about that?
No, we haven't really done that. In terms of the drugs themselves, when in the animal studies, what we've done is we give the rats specific dosing in terms of how much drug that they can have access to. So we wanted to be consistent at first in terms of drug exposure. So we wanted to see the animal escalate within using a specific dose. But other people have actually published papers where they've used higher dosages of methamphetamine than we've used in our study. And other people have used lower dosages, like 1/10 the dose that we're using. There was a paper in 2012 where they were using 1/10 the dose that we're using. But more and more people are using the dose that we're using now, or a little bit higher dose. So we're using this dose to compare sex effects. But in terms of sex effect, there might be potency and dose-dependent phenomenon, but we haven't gone there yet.
Yeah, that would be extremely translatable to human studies, where the dose clearly has is shifting over the years, and may have.
There's no question about it. The drugs are much more potent now, and then we're seeing more methamphetamine-induced psychosis in the United States than we used to have before.
Jean-Lud, let me kind of make another question, change the gear a little bit. We have discussed about shock sensitive and shock resistant, and what are the predictors and potential predictors in terms of who is going to get addicted, who is not going to get addicted, and those sort of questions. I was wondering if you just want to do some sort of kind of self-reflection and go back to kind of your personal history and your personal story, going back to your kind of adolescence, and thinking about your personal characteristics. If you want to do something like a retrospective deep phenotyping in yourself, and thinking about what are the predictors that you think that are, or they were really important for you to be such a successful scientific character in the field. Can you kind of explain some, or think about what are the major kind of environmental or cognitive or even genetic factors that might contribute to their success over the time, and what are the things that people might learn from you in terms of trying to kind of replicate those things in themselves, trying to be able to follow your path?
So those are the type of questions that my psychiatry supervisor would ask. Huh? Anyway, but I think I talk about that actually quite a bit with young people who are trying to go into science. I mean, as I'm a Black American, Haitian American, and there's a shortage of African American scientists, as you know. And so it's a big issue in terms of, okay, I came from Haiti, and I ask the same question to audiences when I'm talking to young Black men and women. okay, what are the odds that I would come from Haiti and then go to Columbia University? I mean, stop. At the time when I went, Columbia was a type 6 medical school in the country. And I did well there and then stayed there as a psychiatry resident. So for me, growing up in Haiti, in a place where I was used to seeing Haitian doctors, black doctors, all of my teachers were Haitian, except for a few Canadian priests who taught in the Catholic school that I attended. Everybody who was quote unquote brilliant and took care of me medically were of African descent. So when I came to the United States, I have some negative experiences. When somebody say, wow, you shouldn't go to medical school, I say, what is this person talking about? Because all the doctors that I know are Haitian and, so it didn't really impact me to the same level that it might have impacted somebody. But I actually, luckily for me, I grew up in a family of upper middle class Haitians. My grandfather, as I mentioned before, is a famous Haitian. So I went to all the right schools in Haiti, and cognitively I wasn't stupid. So that was also very good. So when I attended Columbia, I was able to fit in cognitively, maybe not socially, because my class didn't have too many black students in it. But the other impact is I was able to find, say, I mentioned my scientific director, mentors who said, look, you want to do this, do it and see what happens. And I think it's very important that young people coming from different parts of the country, if they come to the United States, because we do want the brain drain from the other places into the United States to elevate the discussion in the country. If somebody says no to you, you go to the next person, until you find somebody who's willing to mentor you without taking into consideration the fact that you're from some foreign country. So I was lucky in that way. I mean, I grew up somewhat comfortably, went to the right schools. I mean, the school that I went to is considered the best school in our town in Haiti. And my grandfather and mother and father, everybody knew them. So, and then there was never any question that I could do certain things. Everybody knew when I was going to school that I was going to become a physician. But what I didn't know was that I could do research. That was Columbia's. Having attended Columbia was very helpful because that helped me decide, oh, I can do this too, not just take care of patients.
Rita, do you have any conclusion, any final wrap up? Rita, do you have any final?
Oh, I'm sorry. I thought you asked. That was really wonderful. I thought it was really inspiring in so many different ways, starting, everything you said right now about your own history. And then your scientific career going from human neuropsychology, focusing on testing cognitive function in people with addiction. At those times, nobody was thinking that I know, or not many people anywhere, were thinking about cognitive function of people with addiction. So those were some pioneering type of work. And then moving to NIDA and shifting to basic animal type of research. And not many people do that either. So that also indicates a type of ability to explore and ability to put yourself in a entirely novel situation, which you did several times, right? From Haiti to New York to NIDA. Those are major, major transitions in life that I think shows many things, definitely perseverance and persistence, but also exploration and novelty and some sensation seeking, right? So I think we need that as scientists. And it's an amazing ability to do those type of shifts. I think it's very healthy for science, like what you were saying, the diversity of people from different places in the world, but also the diversity of different backgrounds in the same person is enriching their study. That's my conclusion. So thank you, Jean-Lud. That was really inspiring.
Thank you very much. Thank you. Thank you very much.